Hello and welcome back to another video. Today we will learn about enzymes and how they work.
First of all, let's learn about enzymes. In order to fully understand about enzymes, we first need to know about catalysts. Catalysts are chemicals that speed up chemical reactions without being used in it. It essentially just makes the reaction faster without affecting anything else. Enzymes, on the other hand, are biological catalysts that speed up chemical reactions taking place in the body. To give you an example, let's take starch digestion. When you eat food items that contain starch, the enzyme amylase in saliva start breaking down starch into maltose and maltase, another enzyme, break maltose down into glucose which can be absorbed. So, you can see how important enzymes are for the human body. Without them, food digestion.
Some examples of enzymes are amylase, maltase, protease and lipase. Amylase and maltase help the digestion of starch to glucose. Protease helps in the digestion of proteins into polypeptides and later into amino acids, and lipase helps in the digestion of lipids into fatty acids and glycerol.
Now let's learn about how enzymes carry out their function. Enzymes can only catalyze one reaction. This is called enzyme specificity. They contain a surface called the active site to which the substrate binds, forming an enzyme-substrate complex. Now that enzyme bends, or in other words, applies pressure to split the substrate molecule into two. This is called the lock and key hypothesis. When a water molecule is involved in the splitting process, the split ends can never be rejoined. There is another type of enzymes that does the opposite function. They join two.
Lastly, let's discuss about some special properties of enzymes. First of all, we need to know that all enzymes are proteins. Now that we know that enzymes are proteins, we can talk about optimum levels. Just like other proteins, enzymes have an optimum temperature that it works best at. If the temperature decreases, it starts carrying out its function at a slower rate. If the temperature is increased above its optimum level, the particles making up the active site gain too much energy and move, which destroys the active site. When this happens, the enzyme is known to be denatured. The same goes with pH. When the pH increases or decreases from its optimum level, the enzyme's rate reduces and eventually becomes denatured. Do note that a denatured enzyme cannot carry out its task again, even if the pH or temperature is brought back to the optimum level. For example, the enzyme.
Well, that's all about us, and we hope that you would hold tight with us to see amazing content in the future. And join us in upcoming events.
Don't forget to subscribe to our YouTube channel and press that bell icon to make sure that you won't miss out any new content. Also give it a like if you enjoyed it.
Thank you.